Chapter 5. In this section of notes, we're going to be talking about the membrane and a little bit more about how the membrane is built, and then obviously different functions of the cellular membrane and how it helps regulate and maintain homeostasis in the cell. So section 5.1, plasma membrane structure and function. We'll move on to talking about passive transport, so how things can move across the cell membrane without using energy. A little bit on active transport, so sometimes you need to use energy to get stuff in or out of the cell. And then some of the different structures you might find on the surface of the cells and what they do for those. So kind of an interesting way of thinking about this is thinking about capsaicin. And so hot peppers contain capsaicin, and so this is what makes spicy things spicy. And the way that capsaicin works is that molecule binds to a protein in the plasma membrane of the pain receptors in your mouth, and then those trigger pathways within the cell, triggers the cell to open its membrane to calcium ions, and those move into the cell, and then these cause the pain receptors they stimulate the same pain receptors, and that signals a whole other cellular or intercellular pathway that sends a signal to the brain, which the brain then interprets as pain. And it's really neat, I think, to think about something like this physical sensation of feeling this heat in your mouth, really coming down to just like the movement of molecules between cells. I think it's a really cool thing to think about. And again, just gets at we were talked about in the last set of notes. The structure of the cells making up tissues, making up organs and organ systems, and all working together. So how these cells communicate with each other is another major part of that. So the plasma membrane, all cells have a plasma membrane, right? Bacteria, plants, and animals. And it separates the internal environment from the external environment. So obviously you need to keep some things happening inside the cell. You have to separate some reactions from the cell. You want those outside of the cell. Some things have to be able to get in and out, but not everything has to be able to get in and out. And so this is the job of the plasma membrane. There was, back in the day, some kind of like different debate on the different structures of the cell membrane, but eventually we've determined that the cell membrane is what we call the fluid mosaic model, and we'll talk about that. And so the three main components of our plasma membrane: the phospholipid bilayer, which we've already been talking about. And so this has the external surface is the hydrophilic polar heads. So these are the polar heads. And then the inside is the hydrophobic part. They want to get away from the water, right? So they automatically orient themselves inside. So those are the hydrophobic, the fatty acid tails. And then in there we have the protein molecules that kind of float around like icebergs in the sea. I always kind of imagine it like you had like a wading pool full of ping pong balls, and then you also could add like some bigger ball, like some basketballs floating around in there, right? Those would be free to like push the ping pong balls out of the way and move wherever they need to move, and that's kind of like what the proteins are like. They can be peripheral or integral, meaning they could be just on the outside, or they could go all the way through, or on one side, so inside or outside, or go all the way through. So like integral ones goes all the way through. And then we also see cholesterol in there, and so this is again, cholesterol tends to get a bad rap. We think of it as like bad cholesterol, and people have to watch their cholesterol and eat foods that lower cholesterol and things like that. But it does have a natural function, and one of the natural functions is it's part of the cell membrane. It actually helps maintain fluidity, so it sort of like helps break up the phospholipids so they can't pack together so tightly that these guys can't move around. So kind of like the saturated and unsaturated lipids that we talked about in the last set of notes, or in the last unit. These guys make it so that they aren't so densely packed together, so they can allow things to move around. And there's just a bigger picture for you guys to see in your notes. You got the peripheral proteins on the inside. These are integral proteins, and the and individual phospholipid. So some of the other things you might find on the outside of your phospholipid bilayer are the carbohydrate chains, and these help contribute to what we call the cell's fingerprint. Some people call them the 2 Gs because they're glycoproteins and glycolipids. And they help with cellular identification, which obviously is important if you're talking about transplants, right? So this is why you can't transplant, someone who has type A blood, you can't give them type B blood, right? Because those fingerprint things don't match, and then the cell's immune system doesn't recognize the B blood as its own, and so it can attack it. So that's why that's so important. And so two different types of molecules with proteins are glycoproteins, so just proteins with a carbohydrate, and glycolipids are lipids with attached carbohydrate. These guys are on the outside surface of the cell, right? Because it's for cell-cell communication. Again, just a bigger picture of all this stuff together. Peripheral proteins, integral proteins. There's your glycoprotein, your glycolipid. This one's actually attached to a lipid. A little bit of extracellular matrix around here that we'll talk about towards the end of the set of notes. So what are all these proteins that are globbing around in our cell membrane for? Well, it turns out lots of different things. Channel proteins are a major one. These guys right here. They allow for passage of molecules that couldn't normally be able to make it through the membrane, but we still want to get in or out of the membrane. So things can get out that way. There's also carrier proteins, similar to the channel proteins, but these guys actually change shape to help literally escort a molecule through. And then cell recognition proteins, like these glycoproteins that are helping the body recognize foreign substances. You have receptor proteins that bind with specific molecules. So in the beginning we talked about capsaicin. So the capsaicin might bind to a specific protein that is the right shape to accept that molecule. So that's going to allow the cell-cell signaling. Then enzymatic proteins, we've talked about enzymes already, helping to carry out metabolic reactions to speed up chemical reactions. So this little shape thing is using the enzyme to turn into a triangle. And then also junction proteins, so we can actually like bind different cells together in various ways, which we'll talk about later on too. There's just a picture of everything all together, kind of for your benefit. So cell communication first. So signaling molecules are these chemical messengers that allow the cells to send messages throughout the cell, but also to different cells. And so these cell receptors are going to bind to whatever the specific signaling molecule happens to be. So they're very specific, right? This signaling molecule is shaped to recognize this signal. So they're very specific. And once this does bind to it, that's going to initiate some kind of cascade of events that's going to produce a final result. And so these are different no matter whatever your different cell type is. So here we're kind of seeing this as a bunch of different molecules, different shaped things happening, but these could then trigger different events, right? So targeted protein, so you could actually go to a certain protein to get it to do its job. So if it was a structural protein, it could alter the shape or the movement of the cell. It could trigger an enzyme to perform some kind of metabolic reaction, or it could be a gene protein, so it might alter the expression of a gene and tell this gene to start being expressed or this gene to stop being expressed, kind of thing. So these are really important in feedback, both positive or negative feedback loops. I said this earlier, we talk about the plasma membrane as being selectively permeable, and that some substance can get across the membrane very easily, but some things can't. So small molecules, non-charged molecules, those guys can flow through freely. So this is an example here. They're just small enough that they can wiggle their way through the different phospholipids, and they can go back and forth, no problem. Water can also go back and forth, no problem. It is polar, right? So it's not charged, but it is polar. Most polar things can't get through, but because water is so small, it can slide through there very easily. They tend to follow their concentration gradient, which means they move from an area where they're in high concentration to an area of low concentration. So if we're thinking about here, right, you've got none of this molecule inside, lots of it outside, so you have a high amount here and a low amount here, so you're going to tend to go where the stuff isn't, right? They just want to flow that way through diffusion. And so in the end, they become equal, or relatively equal. Moving, we say, down the concentration gradient. Water, like I said, can fit through the cell membrane. Also uses little tunnels called aquaporins that can help speed that process up to really get in there fast. Sometimes they need to be assisted, or molecules or ions need to be assisted by different carrier proteins. Like I said, there's the ion channels that can move charged molecules, and there's carrier proteins that can carry larger molecules. And then some things have to go against the concentration gradient. So going from low to high concentration, these guys do need to use energy in the form of ATP, and we call that active transport. So just another image of this, right? You see these charged molecules are bouncing off. This is a non-charged molecule. It can make it through. Water can go through the aquaporins. This big molecule is too big to fit. It would need to have a carrier molecule help it across. Bulk transport. So if you've got lots of things or a large thing to enter next to the cell, you might use bulk transport. And the two forms of this are exocytosis. So think exo is exit, and this is getting stuff out of the cell. So a vesicle full of whatever this is might eventually just fuse with the plasma membrane and empty its contents outside of the cell, and away it goes. The reverse of that is endocytosis, same exact thing. Now we can imagine if this were something we wanted to be coming in, it's just going to do this process in reverse. And nice little table for you guys to help collect all that information. So chapter 5.2, moving on to passive transport. And so a big example of this is osmosis. This is like super important for cells, right? Regulating fluid concentrations. But when we're talking about this, we're often talking about a solution. And so a solution is going to consist of two different things: the solvent, which is the liquid that does the dissolving. So in most cases it's the water. And then the solute is what gets dissolved. So if we're talking about our example here, we've got a dye crystal, right? This is the solute, water is the solvent. And then diffusion is the net movement of molecules down a concentration gradient from low to high. I say net, and I emphasize net because molecules are always moving both ways, right? So if we're looking at here, we've got this dye molecule, we've got all the water. This shows all the purple molecules moving out this way, but some are just running into other purple molecules and might bounce back this way and bounce back and forth. But overall, as they're ping-ponging around in this fishbowl, they are eventually, overall, a net moving outwards. When net movement stops, we're at equilibrium. Again, they're still moving around, but they're in balance. So that's equilibrium. So the concentration is uniform, so there's no more gradient anymore. And lots of things can affect that that gradient. Temperature, pressure, electric currents, molecular size can all affect the rate of diffusion. Right? If there's a charge, that might affect how charged molecules move. If it's cold, they're not going to move as fast, so it's going to take longer for the diffusion to happen. If they're larger in molecular size, they're also going to take longer to move. So those are all different things that would affect that. Gas exchange is a really common example of diffusion in action, right? When you take in a breath, this is on the inside of your lungs, right? So you've got your bronchioles down to smaller and smaller capillaries into your alveoli. So these little sacs that are surrounded by capillaries, which are really small blood vessels. So you take in a big breath, or any breath, right? And those sacs get filled with oxygen. Lots of oxygen in here, low concentration in your blood, only like 1 or 2 cells thick right here. And so this oxygen can diffuse into your blood just naturally through the concentration gradient. And then what they're not showing is CO2 then is in your blood diffusing out back into the air that you're going to exhale. So osmosis, like I said, is a special case of diffusion. This specifically is the solvent, in most cases water. We're focusing on that moving across a membrane. And so we're going to look at this diagram here. It's going to get smaller in a second, but we're imagining we have this container here. And you look at this percentage is the percentage of solute. So this is like 10% solute versus 5% solute. And so water is going to move across that selectively permeable membrane, and it tends to move it's going to move to where there's more solute. So the solute concentration on one side is high, which means the water concentration is low. And then vice versa on the other side. If the solute concentration is low, water concentration is high. So water is going to move both ways, but the solute can't move. And so you're going to get this net movement of water towards the low water, which is the high solute concentration. I always think of it like the cell wants to try to dilute that solute, right? It can't bring more solute in to increase its concentration, so it has to send water out to dilute it. And so we talk about osmotic pressure that develops due to osmosis if you get stuff moving in and out too much. So look at this diagram again, right? So if you look here, 10% versus 5%, so you have more of the solute, the purple in here, and because there's more purple, that means there's less water, right? There's less water molecules. And so the water molecules are going to tend to diffuse in to dilute these, because these purples can't go out. The membrane is selectively permeable. So over time, you're going to end up with equal amounts of water in there. So when we're talking about this in a membrane, or in a cell, right, across a membrane, we can talk about different types of solutions. And so an isotonic solution is probably the easiest one to think of. This is when water and the solute concentrations are equal. So it's equal inside and outside. So this means water is moving in and out, right? As soon as one molecule of water moves out of the cell, then you're slightly out of balance and another one moves in. So you maintain that equilibrium. So there's no net gain or loss of water by the cell. If we have a hypotonic solution, I'm going to stress that hypo, that means the concentration of the solute in the solution, the hypotonic solution, is lower than inside the cell. And so that means that, remember, the water is going to go where there's more solute, so that means there's more solute in the cell, and so water will tend to enter the cell and cause that cell to swell. In plants, we talk about that as turgor pressure, right? So the opposite would be like being all wilted. So if they're really full, they've got lots of turgor pressure. Animal cells don't have the plant cell wall to help maintain them. So it is possible that they could rupture. We call that lysing. And I think about that, like lysosomes help lyse bacteria cells and break them down. And I always like to remember, like, hypo. Think of some other hypo words you might know, like hypoallergenic means like it's your laundry detergent without all the fragrances. Hypodermic needle goes below your skin. Hypothermia is low body temperature, right? So this is a low concentration. Opposite of that, protozoans have the same problem, but they have these contractile vacuoles that can absorb the water to keep their water balance the same. The opposite of hypotonic is hypertonic, so think hyperactive or hypersonic, right, above. So now the concentration of solute is higher in the solution than inside the cell, and so therefore there's more stuff outside the cell, so water will exit the cell. And so if you're an animal cell, you're going to shrink, which is called crenation, or it's called plasmolysis in plant cells. So the plasma membrane can actually like separate from the cell walls and get all shriveled. The cell wall can't really shrink in size, but this guy can shrivel up. And so there's everything all together. Sometimes you need to do a little bit more to get something across the membrane. So sometimes diffusion just doesn't cut it. I talked about this earlier, right? Small things, the non-polar molecules, non-charged molecules, small molecules, those can move without help. Sometimes you need help, though, so that's facilitated transport. So if you facilitate a meeting, you're helping making the meeting happen. The group facilitators keeping you on track. So this case is transport that's facilitated by the proteins. The proteins are helping it work. And so some molecules are too big or too charged, like glucose, amino acids. Those are both really important molecules that we've been talking about a lot. They got to get into the cell. They can't go right through the membrane, so they need help. And so they can combine with specific carrier molecules to help move them across. So they're still going to go down the concentration gradient, so it's still a passive transport. So you're still going from high to low concentration. No energy is required. So even though you've got a carrier protein that could be changing shape, like this is here, it's still a passive process because it's still going from high concentration to low concentration. Active transport is the other side of that coin. Now we're moving molecules against their concentration gradient from a low to a high concentration. So it's like paddling upriver versus downriver. If you're going upriver, you've got to use a lot of energy, right? Because you're going against the flow. So these guys are still facilitated by carrier proteins, but now we do need to use energy in the form of ATP, adenosine triphosphate. So the classic example of this is the sodium-potassium pump. You need specific concentration gradients of sodium and potassium inside your cells, especially like in nerve cells. This is to conduct a nerve impulse. You've got to keep this balance of sodium and potassium in the right direction. So that usually means like literally pumping some out. And so in this case, we've got a lot we have sodium inside that wants to go out. So the carrier protein is open. You can see it has these nice little square holes to hold these square pegs. The sodium can go in, and then ATP comes along and binds one of its phosphates, releasing some energy. Now we have ADP diphosphate because the third phosphate is there. That provides the energy to change the shape, which allows this guy to open, releasing the sodium. And now this is open with to give access to the potassium, which could come in and bind there. And then the phosphate lets go, and the molecule returns back to its normal position and lets the sodium in. So things to remember, again, it's using energy, 3 sodiums out, 2 potassiums in, and these guys are both moving against their concentration gradient. So in this case, there would be more potassium on the inside of the cell, and so it would want to flow out, but we're trying to pump it in. And we talked about this bulk transport, the exocytosis and the endocytosis. These also require energy. This is an example of these vesicles that are moving around the cell. We talked about this in the last unit with the microtubules in the cell and the little Dynon engines moving along. Those require ATP to be like the train tracks moving the vesicles along. So again, these are going to get rid of large amounts of things. Exocytosis, bring in large amounts of things or large substances. Some different types of endocytosis. Phagocytosis is bringing stuff in, like cell eating large solid material. So white blood cells are going to engulf debris or viruses to help take care of those. Pinocytosis is like cell drinking. So their vesicle is going to form around a liquid to bring in particles that way, or liquids. And then receptor-mediated endocytosis is when it's a specific form of pinocytosis where you have receptor proteins that are in that cell in that vesicle. So only certain things could be coming in. And so here's an example of exocytosis. We already saw something similar to that. Endocytosis, the reverse of that, coming in. Here's the receptor-mediated. Right, you've got these molecules, these receptor proteins here, to make sure that you're only bringing in the correct solute. And so lastly, we're going to talk about what are the things you can do to the cell surfaces. So starting with animal cells first, they have what's called an extracellular matrix. So we abbreviate that ECM. And so this is just a meshwork of proteins and carbohydrates, polysaccharides, that are associated with the outside of the membrane and can do lots of different jobs. So this is the outside here. On this, usually its outside tends to be on top. This diagram is a little bit reversed. And so a bunch of different ones. Collagen, you've probably heard of collagen before, type of connective tissue like a cartilage. So this is going to resist stretching to help that cell keep its shape. Elastin provides resilience to the extracellular matrix. So elastin, kind of like elastic, helps bring things, keeping things together. Fibronectin binds to other proteins, especially these integrin proteins. And so also helping give that cell its structure and its support. So integrin play a role in cell signaling. So integrin is integrated in the protein, but also are usually connected to these different proteins, so initiating the cell cascade, the cellular communication cascades. Proteoglycans, so a long centrally placed polysaccharide. So that's these guys here, right? So you got a polysaccharide in the middle, and then the protein branching off from those. And so these are to resist compression in the extracellular matrix. It's almost like a spring, so to keep it from getting like squished. And these also are assisting cell regulation, cell signaling. There's just a larger picture of everything. And then different ways of connecting cells together. So junctions between animal cells, adhesion junctions. So adhesion, remember, sticking things together. And so you have intercellular filaments between the cells. So two types here. You have the desmosomes. So those are pictured here. So these are internal cytoplasmic plaques. These guys are sturdy but flexible. This actually I don't think was in your note packet. I added this after, so you might want to add that. And so it's a connection that keeps these cells together, but at the same time does allow for some stretching and some movement. So like muscles that are near each other that need to be connected but also still need to move and squish around a little bit, versus tight junctions are these guys here. It's almost like a zipper. These are forming impenetrable barriers, so they're really holding tight. And these are going to be in areas where you want to prevent leakage. So maybe like on the inside of your intestines, right? You don't want your digestive juices that are in your intestines leaking into your body, because that'd be really bad. So tight junctions there are going to form those impenetrable, less movable barriers. Then there's the gap junctions, which help stitch the cells together, but still allow for cell-cell communication. So they have these channels that are joined in between by proteins, and so they're actually still connected. And so messages can pass back and forth, ions can pass back and forth. That's really important in heart muscle, because again, muscle contractions are really just controlled by the release of ions, and so flowing back and forth is going to allow that to happen on its own and facilitate that. So they're still connected together, but they can still communicate very easily. In plants, you've got the plant cell wall, which we've talked about, right? Mostly giving structure. So it's freely permeable for the most part. Obviously, like really big things can't get through it, but it's not really doing much to keep stuff out of the cells, more for structure. Cellulose being the main component, like we talked about in the last unit. Plasmodesmata, similar to the gap junctions, right? These are just like tight junctions in here that still leave like a connection of cytoplasm, so that the cells can still communicate back and forth between each other. So going through the two cell membranes and the cell walls now, so it's a little bit more difficult of a job. And then the cell wall, important thing to recognize, in more woody plants would actually have a secondary cell wall with even more structure, more cellulose fibers, and also producing this lignin, which is that really hard woody tissue. So like if you're cutting down a tree, right? Like most of the wood, or what you think of wood, is a lot of that is the lignin, giving that a lot of strength. And that is that.